by, what do you understand by biological molecules? Any notions?
No.
Any thoughts? Nothing. So, biological molecules are molecules or chemical compounds which are required by the body or by living organisms. Right. So, we have carbohydrates. Carbohydrates are made up of carbon, hydrogen, and oxygen. Right. Then you have fats and oils. They're also made from carbon, hydrogen, and oxygen. And then we have protein, which also has a nitrogen group and sometimes a sulfur group. Okay, right?
Yes.
So pretty basic stuff to here. Now we have when we're talking about biological molecules, we have basic units and then we have larger molecules. So when we're talking about basic units, we have simple sugars. For example, we've got glucose, fructose, galactose, right? And then we have starch, glycogen, and cellulose, which are larger molecules. We have fatty acids and glycerol as the smaller molecules or the basic units of fats and oils. And we have amino acids, which are the basic units of proteins. Okay. Now, how do we test if some food item has these biological molecules? For example, if we have reducing sugars, which are glucose and galactose and all, right? So what we do is we have Benedict solution, which is a solution of copper, right? And we add that to a beaker and we add the solution of the reducing sugars in it. If the solution turns blue to red, right? Brick red, the reducing sugars are present, right? Make sense?
Yes.
Right. And if it remains blue, it means that there was no indication, therefore, there was no reducing sugar present in the solution that we were testing. For starch, you apply iodine, and iodine turns blue black. If it remains the same color, which is normally brown, then you have no starch present. However, if it turns blue black, there is iodine present, right? If a purate, uh, if a solution contains protein, we add burette solution to it. Okay. Now, don't make the mistake of misplacing these two solutions, right? So Benedict is for,
Reducing sugars,
yeah, and burette is for
protein.
proteins, right? So what happens is it turns purple violet when heat is when you add it to the solution, right? The protein solution. If it remains blue, which it's initially blue, right? So that means there was no protein.
No protein.
Yeah. Fats, you have ethanol, right? And you mix it with ethanol and you find a cloudy emulsion or a soapy suspension, right? Which shows that fats are present. This is also called emulsification. Okay. For vitamin
Yes.
C, there is a compound called DCPIP, You add it to the solution and it becomes colorless. If it remains blue, that means there was no vitamin C present. Okay, now let's talk about DNA. What is DNA? DNA, if we break the word deoxyribonucleic acid. So it is an acid, right? So it has an acidic group and it has the first group, which is deoxyribonucleic acid, right? So DNA is deoxyribonucleic acid. This full form is often asked, so you should know the full form as well. Okay, so you have this base, which is a sugar, right? And this is called a pentose sugar because it has 5 sides. Then you have the base, which is one of these, right? A, C, T, and G, which are called adenine, thiamine, guanine, and cytosine. A pairs with T, G pairs with C, right? So you have a pentose sugar, a base, which is either one of these, and a phosphate group. Okay. Now, what happens is you have the structure of DNA. When you have DNA, this thing is made up of sugars and phosphates. Yeah. So this is what you call the sugar phosphate backbone. Okay. Right.
Yes.
So each chromosome is a very long molecule of tightly coiled DNA. So what are chromosomes? Chromosomes are what is found inside the nucleus. They carry DNA. So very tightly coiled DNA is a chromosome. Two strands coiled together form a double helix. Right? So this structure is called a double helix. Right? It moves
Yes.
like so. Yeah. So this is your double helix. Each strand contains chemical called bases, which are adenine, thiamine, guanine, and cytosine. So, what are these? These are called cross links, right? These are the bonds between two bases. A and T are pairs and they form two cross links. C and G are pairs and they form three cross links. Okay, right? You can see that here. You can see that here. So, when you're making right? So when you have two of these, right? So one of them is, let's say, guanine, and the other is cytosine, right? So they have three crosslinks between them or three bonds between them. Okay. However,
Yes.
if you had thymine and adenosine, you would have two bonds. So adenine and thymine. Right, so these have two bonds. Okay. So at this level, you do not need to know the names of the DNA bases. Okay. So this is our chapter for
Okay.
biological molecules. Are you able to continue further?
Yeah, I guess.
Okay. So we're starting with enzymes. Okay. Now, what are enzymes? Enzymes are what are called as biological biological catalysts. Now, if we talk about biological catalyst, the first thing that we should look at is catalyst. What is a catalyst? A catalyst is something that expedites a reaction. Therefore, it speeds up a reaction or it alters the speed of a reaction because it can slow it down as well, right? So, but in the case of enzymes, it is usually okay. So, it can expedite the reaction or slow it down, right? So biological catalyst, a living substance that can speed up a chemical reaction and is not changed by the reaction, right? So this is the second thing, that it remains unchanged after the reaction. It does not get affected by the reaction, right? So it stays the same and it can be used again, right? Now, another property of enzymes is that they are specific meaning they are specific to a certain they are specific to a certain molecule, right? So if an enzyme is working on starch, it will continue to work on starch and it will only work on starch.
Okay.
right? Okay. So enzymes, the way they work is that they lower the activation energy needed for a reaction to take place. So what is the activation energy? Activation energy is the required energy for a reaction to take place. Right? So if the energy required for the reaction to take place is reduced, can the reaction take place faster?
No.
Why? The energy required is reduced. So the energy needed is lesser, right? So it can be provided faster, right?
Oh,
Right?
okay.
So it makes the reaction faster. It is
I'm
essential in all
going to
living
be
organisms regarding the
answering.
reaction. Oh,
I'm going to
sorry.
be answering a little less. So just please continue teaching. I'm listening. There is a problem with the mic of my earbuds, so I'm just going to
I
be
think
answering a little less.
let's leave it here. We can start enzymes in the next lecture.
Okay.
Okay. Let's leave it here. Lafis. Any questions
Okay.
regarding biological molecules?
Nope. No. No.
Okay.